Hello, everyone, and thank you for attending this GenScript-sponsored webinar titled A Brave Approach for Generating Novel AAV Vectors. My name is Dr. Kimberly Arnold, and I'm the Senior Marketing Specialist for Molecular Biology Services at GenScript, and I will be moderating today's webinar. It's my great pleasure to introduce to you today our speaker, Dr. Marcus Davidson. Dr. Davidson is a postdoctoral fellow affiliated with both Michigan State University and Lund University in Sweden. He received his master's in molecular biology and his PhD in neurobiology from Lund University. During his doctoral studies, he worked on developing novel methods for DNA barcoding and applying these techniques to generate AAV capsids with tailored functions in order to study biological functions in the brain. His current research interests focus on DNA barcoding, creating novel AAV capsids, and developing next-generation gene therapies. To ensure that this webinar has high sound quality, please activate listening-only mode in your GoToWebinar settings, or mute your microphone. If you have a question for the speaker, please type it into the chat box in the lower right corner of your screen for us to see. Dr. Davidson will be taking your questions at the end of the webinar. If we run out of time and he is unable to get to your question, we will email his response to you. Now, without further ado, let's welcome Dr. Marcus Davidson to present today's webinar, A Brave Approach for Generating Novel AAV Vectors. Dr. Davidson, it's all yours.
Thank you very much for the introduction, Kimberly, and thank you all for listening. And today I would like to talk about how to use oligo pools to generate new AAV vectors. So basically what we have used is a 90,000 oligo chip from GenScript to modify AAV vectors and how to make them go from this to this in primary neurons, from this to this in human embryonic stem cells, and from this to this in vivo. And I will tell you the full story today, so sit back and enjoy. So my background is in DNA barcoding, and I've worked in two different approaches here. So one is the method development side, and one is the application of these methods we have developed. So for the methods, we have worked a lot with how to generate barcoded libraries and how to sequence these libraries, a task that is not as easy as it seems, and this generated in a publication that in scientific reports. For the applications, we have used DNA barcoding to assess RNA transplicing. It can be found in this publication. To develop novel AAV capsids can be found here on bioRxiv. We also have an ongoing project how to map the connectome of the brains using DNA barcoding. This is still not published. So to just give you a brief introduction to genetic barcoding, a DNA barcode is very similar to a barcode found on any item in the store. It's a unique identifier for a certain DNA strand. And in our case, we've been using a 20 base pair barcode cycled over 3 nucleotides to avoid homopolymers. But if you use any random barcode using 4 nucleotides on each position, the variant grows rapidly, and 20 base pairs will give you 1 trillion unique variants, and that's a number that should be high enough to label any type of cell or cellular process. So basically, DNA barcoding is a very highly efficient method to label DNA. It's sometimes crucial even if you, for example, want to study promoter efficacy, viral capsids, in our case AAV, or if you want to study, for example, transplicing. So in these events, the event leading up to the to the final product is lost, and a DNA barcode is the only way to assess what was happening before the RNA. And another great thing with using DNA barcoding is that it's highly efficient, and in our case we only used 3 rats to screen all of the AAV variants that you will see during this presentation. And it's not a new thing. DNA barcoding has been used in a number of publications. For example, the Adachi used it to map functions of AAV capsids. From Anthony Sater's group, they have used DNA barcoding to map neuron projections, and it could also be used for lineage tracing and different types of screening. And this is a great review from the Sater group that I highly recommend. But today we will talk about our method to generate new AAV serotypes, and we decided to call this method for BRAVE, which stands for barcoded rational AAV vector evolution. So there are a couple of different ways to generate new AAV variants. The most popular is directed evolution, basically where you modify the cap gene with either like error-prone PCR, DNA shuffling between different cap variants, and then you inject or transduce cells, fish out the efficient variants, the ones that could infect. Then you go back and rescreen, and you do this about half a dozen of times. This is a good method to test a lot of different variants, but it's also not reproducible, and it takes many different screening rounds. Another version is rational design, where you have a function that you want to study and you build your vectors based on previous knowledge. This is not as high throughput, and you need to know a lot more about the background of your capsid and your inserted peptide or your modifications to the capsid. So what we wanted to do is to combine these two methods into a high throughput, but still with a rational approach. So what we did in this case was to use the AAV2 vector, modified at the 587 position of the cap gene, and inserted peptides, 14 amino acid long peptides in this position. So we started off with identifying 117 proteins that all had synaptic affinity. Most of them we found from viruses, herpes simplex, rabies, HIV, etc. So proteins or cap proteins that we knew could interact with the synapses. We also picked neurotoxins, neurotrophins, and a subset of proteins that we thought would be interesting to study. So these 117 proteins were digested into 14 amino acid peptides with a sliding window approach. So we used peptide 1 to 14, 2 to 15, 3 to 16, etc. for all of these different proteins. And we then used custom array from GenScript to synthesize all of these oligonucleotides on a chip, and we ended up with 92,918 unique oligos. These oligos were then cloned into the 587 position of the AAV2 cap gene, and at the same time barcoded. So we ended up with 3.4 million uniquely labeled plasmids. And this is the important thing in our method, that we have a connection between the barcode and the inserted oligonucleotide. So what we then did was to produce AAVs in a standard way in 293 T cells. And an important thing is, you can see here on the image that we have moved the rep and cap gene outside of the ITRs. So we have a non-infective AAV, and the link or the identifier that is packaged into the final AAVs you can see on the right is the DNA barcode. So this means that we have generated AAVs free from the rep cap. By doing that, we have removed the serial infectivity, and we could produce AAVs with only 3 plasmid copies per producing cell. And we then ended up with a number of these modified AAV2 capsids. And to start with, we needed to assess how this library was looking. So we needed to link each inserted fragment with its unique barcode. And by doing that, or to do this, we have a new method of sequencing based on a Cree recombinase. So by adding Cree recombinase, we can cut out the ITR, CMV, GFP part and move the barcode closer to the fragment. And by doing this, we can use Illumina sequencing. By moving the barcode and fragment closer, we can use Illumina paired-end sequencing to read barcode and fragment in the same read. And we also don't have to read through the ITR, which is very difficult or basically impossible. So what this sequencing gave us was a lookup table between the combination of barcode and inserted peptide. And so we got one line for each. So we ended up with a data set of 3.5 million combinations of barcode and peptide. What we then was to set up 2 different screening methods, one in vitro and one in vivo. So in vitro in 293 T cells, and in vivo and primary neurons, and in vivo in the brain of a rat. And in vivo screening was performed by injecting the virus in the striatum, and after 4 weeks we waited 4 weeks, and then we dissected out 3 areas connecting to the striatum. So the prefrontal cortex, thalamus, and the substantia nigra. And one important thing to mention is that the goal of this study was to find novel AAVs that had a retrograde transport function. So that's why we injected in the striatum and looked at areas connecting to it to see if we could find viruses that could be retrogradely transported to these areas. And just to show an image of how it looks in vivo using the AAV2 wild type to the left here and our AAV M&M library, as we call it, we can see a completely different transduction pattern compared to the parent AAV2. And this was done by sequencing to assess which capsid went where, to which brain region. We dissected out these areas, and then we sequenced the barcode. So the cap gene is lost. It's a protein on the cap AAV capsid. So the only way to retrieve which capsid it was is to sequence the expressed barcode. So we did that with Illumina sequencing, and we could then build heat maps, which inserted peptide infected a specific area of the brain, or to the left and to the right, which capsid could infect primary neurons or 293T neurons, 293T cells, sorry. And with all this data, we could then start mapping protein function. So in the light blue, in the bottom, you can see the local transduction in the injected site, and dark blue is the transported function of the capsid. So we could start to look at specific hotspots in the proteins, that if a peptide is coming from that area, if we could induce a new function on the AAV. And with 3 different regions and 117 proteins, we ended up with tons of data to analyze to find these unique variants that has a specific function. And so, for example, we can start understanding how proteins work. One example is microtubule associated protein tau that has an area of a microtubule binding domain. But if we pick a peptide from a different region of this protein and put that on the surface of an AAV2 capsid, we get an increase in transport and a new function. And this has previously been shown in publications that certain tau strains can propagate between cells, and we can sort of mimic that function if we put that peptide onto an AAV capsid. And we could also start mapping the efficiency of our entire concept and our screening method. So in black in the middle, you can see the custom array from GenScript with 92,000 unique oligos. Of these, we could clone 90,000 into a plasmid library in the next circle, and the circle outside of that is 66,000, and that's the number of how many DNase-resistant AAV capsid we could generate. And if you look at the dark blue, 17,000 is how many of those that were actually infective in vitro or in vivo. 12,000 of those were found in the injection site, and we found 4,000 to be transported away from the injection site. And this was a good way of seeing that we actually had a working screening system. So what we then did was to pick out 25 of the most interesting variants. So 2 from the in vitro screen and the rest from the in vivo screen. And these ones we then produced one by one, so single productions to start validating them. We also did the M&M null, which is the empty peptide without empty capsid, where we did not insert the peptide. And for controls, we used AAV2-8 and AAV9. And these were all produced with a self-complementary AAV GFP, driven by the CMV promoter. So we started by validating these in vitro. And we did this in 203 T cells and primary neurons, the same where we screened. We also used primary glia from rat and human embryonic stem cells. So looking at the first thing we did was to see that we could produce all of our capsid in a fairly efficient way. And you can see here, this is one production round, so there are no error bars. But you can see in green, we have the AAV2 wild type, and most of them could be produced with the same or higher efficiency than the parent AAV2 wild type. And this is basically showing the same. In blue, AAV2, and to the right, we have our different versions of M&M capsids, that we call them. And once again, we could produce them with higher efficiency. Then looking at the in vitro validation, we can see that AAV2, it's the top row, second from the left. AAV2 is fairly good at transducing 293 Ts. And the one we picked from our screening was this M&M 026, and we can see that we The one we screened for to be best in 293 T cells is actually performing quite well. And it's more obvious when we look at the primary neurons. You can see the AAV2 wild type is not very efficient, top row, second from the left, but with a modified version, M025, we can transduce a lot of the primary neurons, maybe not all, but it's highly efficient. We also looked at primary cells from rat, primary glial cells, and we have AAV2, 8, and 9 on the top row, plus the empty one. None of them are very efficient, but in the bottom row you can see that M010 and 26 are very efficient when it comes to transducing glial cells. And looking at the human dopamine neurons, the embryonic stem cells, 2, 8, and 9 to the left are not transducing at all, but by putting a peptide on the capsid, AAV 8, 10, and M&M 8, 10, and 26 are highly efficient of transducing these human ES cells, which was, of course, nice to see that even if we screened in a rat, we can see transduction in human cells. And that is, of course, important in the AAV field, that you have translation to that it can work on human cells as well. We also, the main part of this project was, of course, the in vivo part, and we validated all of these picked the selected variants in vivo. And so this is the injection site, the stratum, and we can see that we have transport to the prefrontal cortex and to the thalamus. So the capsids that we screen for behave in the same way when they are produced one by one, which of course was an important thing for us. And this is seen a bit clearer here, where we compare the AAV2 wild type with our M004. We have transduction. The AAV2 wild type stays in the injection area in the stratum, but with the 004, we have transduction from the prefrontal cortex to the thalamus, amygdala, and all the way back to the entorhinal cortex, which is probably a bit clearer to see here. So adding a 14 amino acid peptide makes a massive difference to how the AAV behaves. This can further be seen here with the same vector M004. We injected mCherry and GFP in the stratum. So left side mCherry and right side stratum, and we can see the prefrontal cortex expression. We can see the thalamus and the amygdala, and all the way back to the entorhinal cortex, making this one of our useful vectors for future studies. One of our favorite vectors is the 008, and this one has unique function that it can be retrogradely transported to dopaminergic neurons in the substantia nigra, and these cells are normally very difficult to transduce using a retrograde approach. And what's interesting with this variant is that the peptide displayed on this capsid is coming from the canine adenovirus, the CAV. And this virus, the canine adenovirus, has previously been shown to have a retrograde transport and to be able to transduce dopaminergic neurons. And when we take just one peptide from this canine adenovirus and put it in an AAV, it behaves the same way. So, and that is a very interesting observation, that we can map protein functions by this approach. And this is exemplified here with the flexed vector injected in the stratum, and we can see highly transduced area to the right using 008, but basically no transduction using an AAV2. And this vector, we also did a fluorophore versus functional protein approach, where we injected a tag BFP, synuclein, or M-cherry, synuclein in green, in the stratum in 3 different locations. And then we looked at the transport to substantia nigra, which you can see here. And doing these in serial, you can see the M-cherry, the tag BFP, and then in green, the alpha-synuclein being transported to dopaminergic neurons in the substantia nigra. We also did an approach to see if we could transduce transplanted human cells, human embryonic stem cell, with the dopaminergic differentiation. So these cells were transplanted, and then we injected our 008 to the left and 004 to the right. And so these cells were transplanted into the nigra, and we could see that injecting this vector into the stratum, we had a retrograde transport with both these vectors, and they were targeting these human transplanted cells. So another validation that it works both on rat and human tissue or human cells in vivo. We also did performed a functional study to see that we could use these vectors for a functional approach. So we injected 004 in stratum and a flexed dread receptor, an activating dread in the basolateral amygdala to study anxiety. So basically only the cells in amygdala projecting to the stratum would have a functional dread because of the transported CRE. And in B and C, you can see the dread group to the left, and in B, and in C, the control group. And we can see that the control group spend much more time in the open arms than the dread, suggesting that there is an increased anxiety in these, and validated by staining in F and G of the receptor. Another interesting vector is the M&M017. And this in this capsid, we have the peptide originating from tau, and we can see that in B, and that this one is efficient in transducing microglia and in astrocytes, or it can transduce those. And in C, we have human astrocytes in vitro, and compared to the M&M002 to the left, which was good in primary neurons, but it's not working on glia, but M&M017, we have quite good infectivity in human cells. And in D, we did quite an interesting experiment where we wanted to see if we could compete out the vector using 3 different tau species, so T44, T39, and K18. And by adding these proteins together with the M&M17 vectors, we could see that either an increased uptake or a decreased uptake of the vector, so suggesting that there is either a blocking or a co-uptake of this AAV vector into the cells. So to summarize our BRAVE screening approach, we could perform this in a single screening round, and this is fairly unique to AAV vector screens that normally requires 5 to 6 screening rounds to find the functional capsid, but we could find 25 novel capsids with our single approach. And of course, this is interesting from a 3R perspective if you want to reduce or refine, replace your research animals, and it saves both time, money, and animals, which I think is a very good thing. We use mRNA as readout, so that means we don't need a lot of infectivity. We can also use the number of mRNA, the abundance, as a readout for efficiency of our vector, and we have no only the AAV vectors that infect and make it to the nucleus and start transcription are those that will be analyzed. So we are not looking at vectors being trapped outside of cells or inside cells that don't make it to the nucleus, which is the case in for example, directed evolution. We found 25 novel serotypes that we have continued validating. And we can see that the protein or the peptide function from the original protein, or in many cases, in our case, in this study, from viral capsids, that the protein or peptide function is reflected in the AAV viral function that we can see. These protein motifs that we found can then be mapped using a hidden Markov model called the hammock. So we can start looking at unique peptides, or in unique positions, amino acids in unique positions that are crucial or detrimental for the function of the inserted peptide. We believe that this method can be adapted for any AAV serotype, or any virus type for that matter. And there are, if you go a bit further and using DNA barcoding and this type of screening, it can be used for many different other applications. And oligo synthesis is no longer a limiting factor. When we started this approach, it was they were fairly expensive and could not be produced in the numbers we wanted. But nowadays the price has gone down, quality is going up, and it's not the limiting factor for to do this kind of large screens of viral capsids or whatever you want to do. And also, to quality-wise, quality is good nowadays, but even if it's every now and then a mismatch or a mutation in your synthesized oligo, that could just increase your pool of variants you want to test. And I did not do this work myself. I would like to thank all the people in Lund involved in this, and especially my mentor, Thomas Björklund, and also Patrick Gangwang, Andreas Hoyo, and Morgan, who was a big part of this project, and of course all the funding sources for making this possible. And I'm just going to end with a fly-through of a rat brain using the M&M004 injected into the striatum. And thank you very much for listening.
Thank you, Dr. Davison, for such a wonderful presentation. We'll now move on to the Q&A portion of this webinar. Please take a few minutes now to type your questions into the question box. While you're typing in your questions, I'd like to introduce GenScript's oligo pool synthesis service that Dr. Davison has utilized in his presentation today. GenScript's oligo pool synthesis service provides a perfect solution for cost-effective library construction and efficient high-throughput screening. It's ideal for the creation of large, diverse libraries, including CRISPR guide RNA libraries, mutant libraries, and also for NGS targeting libraries. Using our advanced semiconductor-based synthesis platform, we're able to precisely synthesize thousands of user-defined oligos simultaneously on a cover slip-sized chip. The electronic control of the synthesis process produces high-fidelity oligos and ensures only the sequences you designed are represented, which leads to faster identification of your hits and minimizes your downstream screening burden. Our 2 chip sizes allows the creation of any size pool to meet your experimental needs. In addition, we guarantee the final yield of synthesized oligos, ensuring you'll have 100% downstream amplification of each oligo and be able to create diverse libraries. If you would like more information about our oligo pool service, please visit our webpage listed here. In addition, GenScript offers a number of downstream services to complement your oligo pool, including cloning, plasma prep, and library construction. For more information, please visit our website at www.genscript.com. All right, well, with that being said, we will start our Q&A session. Dr. Davison, the first question we have is in regard to the BRAVE screening. How long does it take to perform a screen like this?
That is a good question. The actual screen itself is about, I would say, 4 to 5 weeks. Most of the work or the time-consuming part is to identify your proteins that will then generate these peptides that we insert in the capsid. So that bioinformatics or that search has to be done manually. So I would say that's the most time-consuming part. Cloning and AAV production, and then the screen, maybe 6 weeks, and then add the bioinformatics before.
Okay, and is there any upper limit of the variants that you can screen for?
I would say no, not really. It depends, of course, on the how much funding you have, how many oligo chips you can buy. But if you have a decent amount of money, and then you can definitely generating the library is, I don't see any upper limit. So we made about 3.5 million barcode plasmids, but you can easily go higher than that. So I would say no.
Okay. Have you seen any MNMM capsid that make the AAV vectors go only retrograde, meaning no local transduction at the injection site, but just specifically retrograde?
That is it's a good question, and it's like 100% only transported and not found in the injection site. I would say no, but we have those that are more efficient in being taken up than they are transducing the injection site. But I think if you deposit an AAV in the brain, it will, I think in all cases, infect some cells. Then the retrograde transport could be more or less efficient, if that answers the question.
Okay. And is it possible to identify any synaptic jumping AAVs using this screening method?
Well, I haven't seen any AAV so far that can jump a synapse. So if someone has one of those capsid, it would be interesting to see it. No, we haven't seen any synaptic jumping. But if the person who asked this would like to give some more information, I would be happy to look into that.
Okay. And can you explain again how you tie the barcoded DNA with the peptide sequence during oligo insertion?
Could you repeat that one, please?
Can you explain again how you tied the barcoded DNA with the peptide sequence during your oligo insertion?
Yes, definitely. Can I share my screen again, or maybe not?
I guess just briefly explain it.
So in the plasmid, we have the AAV rep cap and inserted fragment, and then in the same plasmid we have the ITR containing CMV GFP barcode, and that's where we did a Cree. We treated the plasmid with Cree to cut out the promoter and GFP to bring the barcode closer to the inserted fragment, and also to get rid of the ITR, since you cannot sequence over that one. So basically, it's just a paired end sequencing, one read from the barcode side and one read from the inserted peptide side, and then you get the link which barcode is sitting with which fragment. And there is more information about this in the paper that we put on bioRxiv.
Okay. And when you were cloning the oligos into your plasmid library, what method did you specifically use for that cloning?
We tried several methods, but we found that Gibson assembly was by far the best method.
Okay. And roughly how large is the barcode that you've used, and how does that affect any of the functions or the processing downstream?
So we used a 20 base pair barcode with a cycling of the nucleotides to avoid homopolymers. You can go longer or shorter depending on the need, but a 20 base pair barcode gave us enough diversity. If you don't think it's enough with 20 base pairs, you can definitely go larger. I mean, yeah, you might actually be unlucky to, let's say, create a microRNA target site with your randomized barcode, but the only thing that will happen is that you will lose it. So it will just be, I mean, if this happens, it will be targeted and degraded. So it's not going to create any false positive. You might be unlucky to lose one or two, but I think it's highly unlikely.
And why did you choose to use AAV2 as a starting template, but not other AAV capsids such as AAV9?
That's a good question, and AAV9 is extremely hot at the moment. But we wanted to do, so the entire screening was based on retrograde transport in the brain. We decided to use AAV2 because it's the most studied, and it's also free to use. AAV9, I believe, is still under patent. And it just seemed like a decent capsid to start with, but you can do this with any of the AAV variants out there.
Okay. And can this library be tested with other types of applications, doing IV injections or muscle injections?
I guess this is still on the AAV9 topic, and yes, you can definitely do it. I think it's, I mean, we haven't tried it yet. We would be happy to collaborate with if someone wants to do it. It's also maybe not really fair to our library to test that IV, for example, because it's designed to be retrogradely transported in the brain, and that's how we chose the proteins and that then became the peptides. So they were designed to target or to have this synaptic affinity. I think if I was doing an IV, I would probably redesign the library and generate a new one. But since we have this one ready, it's definitely worth testing it.
And can you do both positive and negative selection with this?
No, we cannot do. Yes. We can actually now. It's possible to do it with the new sequencing approaches out there. But we are working on another version of this that is a bit more elegant and that will allow for positive and negative without using very expensive sequencing methods.
Okay. And then this is a two-part question. Have you tried alternate insert sites such as amino acid 453?
No, we have only done it at the 587. For a reason that this is, with this approach, with the sequencing, the paired end, when you link the barcode and insert the peptide together to see your library, you need to have them in, like, you need to be able to do this Cree approach, and it was much easier to put it in the 587 position for this. So no, we haven't tried any other insertion sites yet.
Okay, and then so do you use linkers to bookend the peptides themselves?
We tried a few different linkers. That's more information about that in the paper also. We had 4 different linkers: 1 alanine, 5 alanine, a G4S, and I cannot remember the 4th one. We found that 1 alanine on each side was, we got more positive hits on that one. So I think that one would be the next for me to use the next time.
And did you find that the peptide that only transported to the nigra, but not the other areas when injected in the striatum? Was there any peptide that did that?
We have the 008. It's by far the best one I've ever seen to go to the nigra when injected in the striatum. I cannot say that it will not go anywhere else, but it has it has a preference for those dopaminergic cells projecting to the striatum.
Okay. And one final question: is it possible to get a hold of your library or selected capsids?
Definitely. It's we would be happy to collaborate. So just send me an email and we can work things out.
Okay, great. So we are actually out of time and have to conclude this webinar. Once again, any responses to the questions that we were unable to answer will be emailed to you directly. And thank you, Dr. Davidson, for sharing your research with us, and thank you everyone for taking the time to join us today. We'll see you next time.